Okay, so for study session 5, which is still under enzymology, study session 5 basically about enzyme inhibitions, where we talk about enzymes playing a very important role in many biological process. And then understanding the mechanisms of the catalysis is very important. And how does that understanding of the mechanism how an enzyme catalyze reactions comes about, or even how an enzyme is discovered to play an important role in a particular reaction when a particular pathway was able to be discovered. This was achieved through understanding the effect of inhibitions of some certain enzymes. So a particular enzymes can be inhibited, and then after inhibition, then study the substrate that accumulate, then also study which pathway is affected. That is how enzymes, really, or the process of enzyme inhibition was used to identify within the functions or which pathway in particular or which of the action a particular enzyme is involved in. And then we talk about the mechanism of reaction. How does this enzyme specifically convert a substrate to a product? The mechanism that is done is achieved by the concept of inhibition. So what is this enzyme inhibitors? Because the inhibition is not possible through molecules we call inhibitors, enzyme inhibitors. These enzyme inhibitors are nothing but compound that bind to an enzyme, either the active site or a regulatory site, thereby preventing the substrate from binding and converting or stopping the conversion of substrate to a product. Remember, we talked about an active site where an enzyme binds to a substrate and then that leads to specification of events which give about conversion of the substrate to product. So inhibitor of an enzyme will be very interact with this site, either it's the active site or another site we call a regulatory site, usually called allosteric site. So they interact with this, and then when they interact, they either interact at the active site and prevent the substrate from binding, or they interact at the regulatory site, thereby once they attach to the regulatory site, they cause some conformational changes that will affect the accessibility of the active site, thereby preventing the substrate to binding, or even when the substrate bind, upon binding of the inhibitor to the regulatory site, will inhibit the sequence of event that leads to the release of the substrate that was already bound, or the release of the product that was already bound in the active site of the enzyme. So before we talk about that, we made mention of two types of the interaction that can occur between an inhibitor and an enzyme. The first one, we have a reversible inhibition, or we have irreversible inhibition. And we know when we say reversible and irreversible, the situation whereby the binding can be reversed, and the situation whereby the binding cannot be reversed. In a situation where the binding cannot be reversed, the enzyme, or sorry, the inhibitor actually binds to the either the active site or the regulatory site through a kind of an interaction, usually kind of reactive modification, whereby the functional group of the inhibitor will bind to the some amino acids, so through a kind of a very strong covalent interaction. And this kind of covalent interaction will be permanent, such the enzyme will be inhibited permanently. So usually this kind of inhibitions called swing site inhibition, and molecules that does that in most cases even considered biologically poisonous because they have the potentials of causing a lot of havocs. But for the irreversible inhibition, that is what was actually used to even understand like the mechanism of the reaction, understanding the role, the actions of an enzyme or the reaction which an enzyme specifically plays an important role, or the pathway in which an enzyme plays an important role, as well as the mechanism through which that enzyme performs its function. So reversible inhibition, like I said, it's the one aiming to the interaction between the inhibitor and the binding pocket is reversible. And this has been exploited in designing drugs inhibitors. Some enzymes are critical in understanding the role of an enzyme in some pathological conditions. So reversible inhibitions were classified into two. We have competitive inhibition, we have non-competitive inhibition, we have non-competitive inhibition. So we will talk about the different type of inhibition. Remember, we're talking about I think there's a point where we talk about, let me think, look back there. Okay, we talk about the functions of synthetic nucleotide, where we say synthetic nucleotide play an anti-cancer role, antiviral role, and we made mention how this synthetic nucleotide actually play important role as antiviral or anti-cancer. How is that possible? We mentioned that some of those enzymes that involved in DNA synthesis, the DNA polymerase, or that involved in transcription, DNA RNA polymerase, we use these nucleotides. And these nucleotides, you can synthesize them. And this synthetic nucleotide, sometimes you can synthesize a nucleotide that is a bit different from the normal nucleotide. For example, thiamine monophosphate or thiamine monophosphate. So thiamine monophosphate can be modified slightly in which then, for example, the nitrogenous base may contain an additional oxygen or may contain an additional OH group. That OH group that made it to be a bit different from thiamine monophosphate. So it's like hydroxylated thiamine monophosphate, and that will make the molecule now, the thiamine monophosphate, to be more reactive than the initial thiamine. So if the RNA polymerase or DNA polymerase in this case will take thiamine monophosphate, there is the hydroxylated thiamine monophosphate, there is tendency that this more reactive molecule may tend to react with the active site of the DNA polymerase, and when it reacts, it will not be able to incorporate it in the process of DNA synthesis. So once it remains there, the enzyme cannot add it to the growing DNA. Therefore, it will block the binding site. No one will come in, and it didn't go out. That is atypical inhibition. And when that happens, we explain the cases of antiviral and anti-cancer cases with explain them. So classically, also you have most of what is happening here. We can have some compounds designed to bind to the active site of some enzymes. So basically, while designing a compound that binds the active site and prevents the reaction from occurring, you will see that the molecule that is designed must have to be analog, or must have a structural analog, must be similar structure-wise with the compound, so with the substrate of that particular enzyme. Thereby, once it's react with the active site, it will not be released, so it will remain bound to the active site, or it can competitively prevent the substrate from coming in or binding to the active site, either it with respect to concentration or with respect to reactivity. So, for example, we have typical some of those inhibitors that has been used clinically to treat certain conditions, disease condition. For example, the use of penicillin in treating as an antibiotic. As an antibiotic, we know we talked about glycosaminoglycans when we're talking about structural polysaccharides. And we say the glycosaminoglycans were typical examples of structural polysaccharide, and they form a component of bacterial cell wall. How do they form a component of bacterial cell wall? We made mention that this glycosaminoglycans polymer is again linked or cross-linked by a short peptides. And these short peptides were synthesized by the action of transpeptidases. So this enzyme transpeptidases, if you have a molecule or you design a molecule that inhibit transpeptidases, then this cross-linking process will not occur. And the cross-linking is what make the peptidoglycan, the cell wall of bacteria, become very strong, thereby preventing the movement of water and other substances into the bacterial cells. So, but if this cross-linking could not occur, meaning water and other things can diffuse into the bacterial cells, and the bacterial cells will, of course, take off a lot of water, and this could lead to bursting of the bacterial cells, which kill the cells. So also we have disulfiram. Disulfiram is an inhibitor of an enzyme called acetaldehyde dehydrogenase for alcoholism, to treat alcoholism. Those people taking alcohol, we know that the ones who consume alcohol will be converted to acetaldehyde in the biological system. So first of all, we have the action of alcohol dehydrogenase. Then alcohol dehydrogenase will convert the alcohol to acetaldehyde, and then acetaldehyde dehydrogenase will convert acetaldehyde to acetates, which is then being converted to acetyl-CoA, and then it is used to synthesize fatty acids. So I remember when we talk about the formations of fatty liver, deposition of fatty acid in the liver cells, get to fatty liver with respect to alcoholism. So what happened is the disulfiram actually bind to the active site of acetaldehyde, thereby preventing the formation of acetate from acetaldehyde. So definitely when you take this enzyme, is when you take this compound or a drug, will definitely inhibit the formation of acetates. Therefore, you will have accumulation of acetaldehyde and you have accumulation of alcohol. For those people that are taking alcohol, will have high concentration of acetaldehyde and alcohol in the body system. And this is very dangerous and it makes them feel sick. Therefore, once you take alcohol, if you have the effect of this or you have this acetaldehyde dehydrogenase inactive or inhibited by disulfiram, you fall sick. So people that fall sick after taking alcohol will definitely have to abstain from or run away from taking alcohol. Another example is aspirin, which is a non-steroidal anti-inflammatory drug, and we know that it inhibits cyclooxygenase. Remember, we talked about cyclooxygenase, and we talked about this aspect of fatty acid metabolism. Synthesis of eicosanoid, where we talked about eicosanoid being a dry or generated from the 20 carbon polyunsaturated fatty acid, arachidonic acids. And then we have prostanoid, we have linear eicosanoids, and we said these eicosanoids then leading to formation of prostaglandins. So these prostaglandins, they are involved in the inflammatory signals, contraction, relaxations. This is related to pain. Shown quite a number of prostaglandins, etc., etc. So they are basically involved in inflammation. So inhibition of this will reduce inflammation or reduce contraction and relaxation of smooth muscles and bones. Hence, we know that pain is involved, physically pain involved in contraction or relaxations of muscles. The typical reasons behind pain. So aspirin, this is what aspirin do actually. It's to prevent cyclooxygenase. So then going back to different types of reversible inhibition, I mentioned competitive inhibition, uncompetitive inhibition, and non-competitive inhibition. Now to summarize, you've already written, you must have written on these different types. You have competitive inhibition where the inhibitor competes with the substrate for the binding site of an enzyme. So the inhibitor now binds to form the enzyme inhibitor complex. So what happens is these competitive inhibition, the inhibitor will compete for the binding site. So if you have the inhibitor even in high concentration, it can displace the the substrate. So therefore, we will have the inhibitor binding to the active site to form inhibitor enzyme inhibitor complex instead of enzyme substrate complex. That is competitive inhibition, where they compete between each other, the inhibitor and the substrate. While in uncompetitive, there is no competition between the inhibitor and the substrate. Rather, the inhibitor binds to the enzyme substrate complex, thereby preventing because we know that in enzyme catalyzed reaction, we have at this step, we have the enzyme and the substrate coming together to form enzyme substrate complex. So you have the substrate binding to the active site of the enzyme to form enzyme substrate complex. So the enzyme substrate complex will dissociate. With dissociation, you have the substrate already been modified to a product. So it dissociate to product and an enzyme. The enzyme will remain unchanged while the products will change. So what was released has been changed. Initially, it bind as substrate, it was released as product. So in the case of uncompetitive inhibition, what you have is the enzyme, once the substrate can bind to the active site because the inhibitor doesn't bind to the active site, it binds to a different point we call allosteric binding site or a different binding pocket. So this is bind to the different binding pocket. Once it binds to the different binding pocket, it prevents the substrate from dissociating in form of product. While in non-competitive inhibition, we have it bind to either the free enzyme, where you have the inhibitor will bind to the free enzyme, or it binds to the enzyme substrate complex, or binding to a site different from the active site. So in this case, you have a different site binding pocket. So once it binds, it can prevent the binding of the substrate to the active site. So it binds, it prevents the active site through inducing some structural modification, which inhibits the substrate from binding to the active site, or rather even bind before the substrate binds, so that the substrate will not be able to bind, or bind in such a way that the substrate is already bind to the active site, but it will not be dissociated. So in either case, you will have either enzyme inhibitor complex or enzyme substrate inhibitor complex. Okay, the fourth thing we have enzyme regulation. We said that enzymes has been used for regulatory, as a regulatory molecules to regulate the different pathways which reaction take place, which reaction does take place, which one is active, which one is inactive. So we said enzymes can be regulated through two major processes. We have allosteric regulations, we have covalent modification. Some of you write on allosteric regulations, while some write on the covalent modification. Now, in summary, what is this allosteric regulation? We say in allosteric regulation, it's involved the binding of a molecule to a pocket other than the active site, which we just mentioned, with a resultant modification of the activity of the enzyme. So you have the molecule bind to a site on an enzyme other than active site, which we call allosteric effector or modulator or modifier. So the molecule will bind to the allosteric site. So the binding to the electric site will now tell the active site or modulate the active site, whether the active or the activity of the enzyme to increase or decrease, or even completely inhibit the enzyme. So this molecule will bind and then causes the sequestration of changes in the activity of the enzyme for the allosteric regulators or allosteric effectors, allosteric modulators, allosteric modifiers. Then when this allosteric effect to enhance the enzyme activity, increase the enzyme activity in cases, we call it allosteric activator. But when the interaction leads to decrease in the enzyme activity, we call it allosteric inhibitor. A typical example in this case is end product inhibition, what we call feedback inhibition. Feedback inhibition is when you have For example, we have a product of a particular partner between one of the first enzymes in that pathway. For example, when you have E plus B catalyzed by X over C, and then C plus D catalyzed by Y equals D. You have D plus E catalyzed by C equals F. And now you have F would be an allosteric inhibitor of X, which is the first enzyme, or of Y, which is the second enzyme in the process. So you can have F if the F accumulate, or if A and B react to give C, you'll have high a lot of C. So C will definitely react with D on and E will also react with F on G, you say. Then which we say is catalyzed by C. So the accumulation of the final product in this case, which we said G. G can accumulate and then inhibit the first enzyme. Let's see, the first enzyme will say X, it can inhibit X. So G can be an allosteric inhibitor of. So when you have G binding to C, binding to X, X, which is the first enzyme, or binding to the second enzyme, which is Y, it can bind to an allosteric site and then causes inhibition. This is what we call feedback inhibition. End product, because it is the last product that is being produced in the pathway, that is why it is called feedback inhibition. So the end product inhibition, or typical example of feedback inhibition. Then in covalent modification, this involves the conversion of an enzyme from active site to inactive form, or from inactive form to active form through covalent modification. This modification usually occurs to phosphorylation reaction by the action of enzyme called kinase and phosphatases. A typical example we give is phosphorylase. Phosphorylase, glycogen phosphorylase is an enzyme that involves synthesis of glycogen. It can be active site, it can be inactive site. The active site usually is phosphorylated form. It's covalently attached to a phosphate group that made it to the active. So if you want the enzyme to be inactive, then you remove that covalent interaction with phosphate to, by action of an enzyme called phosphatase. So phosphatase will click the phosphate attached to the phosphorylase. So once you have the phosphate attached to it, it's active, so now it's called phosphorylase. But if you have an enzyme called phosphatase, then you can remove the phosphate group into inactive as phosphorylase. So phosphorylase B also, if the need to be, the body needs to synthesize the glycogen to break down the glycogen, then another enzyme will attach the covalent bond to the phosphate group. These are called kinase. Now we phosphorylase B to bond phosphorylase B, which is phosphorylated phosphorylase B will be phosphorylated. So you have the phosphate group attached to, and then it will become active. Another example also is hydrolytic cleavage of the covalent peptide bond, as in the case of what we call zymogen in digestive enzymes like trypsinogen. Zymogen is inactive trypsin. Then you have a cleavage of some peptide bond. Removal of some of those peptide bond leading to formation of trypsin. We have trypsinogen converted to trypsin, chymotrypsin converted to trypsin, etc. All this is regarded as covalent modification. Now, another example is that is that, and then the clinical applications of enzymes, which we have already discussed in the previous. I think if you remember this, we talked about the isozymes of lactic dehydrogenase, where we say it LDH1 is a very important in kidney a heart and kidney red blood cells, the LDH3. Then we mentioned also AST and ALT hepatocyte, which tells about the liver function test. Then also cases of acute pancreatitis, we check the presence of lipase or alpha amylases in the blood. So if you see those, like we just explained the purpose of where we made the mention of how differentiation bring about some activation or the activations of some certain genes in a different cells, depending on specialization. Cell will specialize, for example, to involve in transport of oxygen, like red blood cells. So the red blood cells need for the transportation of oxygen within the blood. So the protein involved in the synthesis of hemoglobin will be expressed, and you have hemoglobin being synthesized cells. And there are many other things that will not be needed. So those genes will be shut down, but they are not needed. Likewise, for example, in muscle cells. In muscle cells is not involved in transportation of oxygen. So you will not have the globin genes, the hemoglobin genes being expressed there. Then what is needed, you have skeletal muscles, basically, or smooth muscles, basically involved in contraction and relaxations. And there are the main molecules that are needed there. The actin myosin, the contractile proteins, or the skeletal proteins, or the structural proteins that are involved in contraction and relaxations. So they are needed as such, their expression will be needed there. So likewise, there are some certain enzymes, certain proteins that are being expressed in some certain cells depending on the function of the cells. And of course, we know that a specific type of cells come together to give tissue, specific come together, tissue to form an organ. So you can have this in such a way that a particular organ will have more expressions of certain proteins. Likewise, the same enzymes are proteins, so you can have some specific enzymes being found highly expressed in some certain organs, while in some certain organs they're not expressed. Even the isoforms of this is high in this, while in this you have an isoform of this. So the concentration of those enzymes in blood circulations can tell about the condition of that organ. We'll give an example of the AST and the ALT. Some of you that are working in the clinic, in the hospitals already know that tests like AST and ALT are meant to check for the liver function. And of course, why AST and ALT liver function, meanwhile, you can have some of these things and also express in some certain organs simply because in the liver, you have the high expressions of this. So if you want to see the concentration of AST and ALT in blood circulation, it simply tells about the condition. So likewise, we mentioned LD metric hypotenuse for head, kidney, some specific isoform like LDH1 for heart, like LDH3 for kidney and brain, like LDH5, lactic hypotenuse isoform 5, liver skeletal muscles. We made mentions of alpha amylase and lipases, which give a direct translation of problem that comes from the pancreas, for example, acute pancreatitis. And then we know CK, protein kinase, that is being used clinically in the hospital to tell about the condition of the heart. So basically, this is what I need to advise regarding this.